None of this should exist.
Tube worm fields. The submersible window fills with white towers, standing like a frozen, bleached forest under a sky that never existed. When the team descended, they expected a wasteland of pumice and mud. They found a biological impossibility. Instead, these living towers are anchored directly to the rock, acting as skyscrapers for bacteria that manufacture food from toxic chemicals seeping out of the Earth's crust. They don't need sunlight. They don't even know what the sun is. The microbes turn methane and sulfide, gases that would instantly kill a human, into raw sugar. This is chemosynthesis, and it is the exact reason scientists were completely stunned to see them thriving at over 31,000 feet. We aren't looking at a smattering of microscopic extremophiles. We are looking at chunky, highly structured, multi-story communities existing under a physical weight that would compress a military submarine into a crumpled soda can. The established oceanic models dictated that only scattered scavengers should pathetically eke out a living in this abyss. The evidence out the window shows a roaring, high-density metropolis. The bacteria living inside the towers run the chemical refineries, and the tube worms provide the real estate. That single relationship builds an entire localized economy. Tiny, pale scavengers graze on the bacterial runoff. Larger animals arrive to eat the grazers. Submersible footage captured this entire food chain operating flawlessly where photons go to die and physics actively vetoes biology. Biochemists can explain the basics of how special fats stop their cells from imploding. What they cannot explain is the logistics of how these dense communities assemble in the first place. Did larvae just happen to drift down, miles through the freezing dark, and accidentally land on a microscopic chemical vent? Or did ancient lineages slowly crawl down the trench walls, adapting over millions of slow, punishing years? The geneticists keep finding tiny tweaks in deep-sea genomes, but for these tower communities, we still lack the master blueprint. If a sprawling metropolis can exist in the absolute Hadal dark, the real terrifying question is what else is hiding just past the lights of the submersible. Chemosynthetic clams. As the rover's lights pan across the mud, they illuminate rows upon rows of pale shells, half buried like a graveyard. Except, these clams aren't here to casually filter plankton like their coastal cousins. Packed inside their gills are armies of bacteria that metabolize the caustic chemicals hemorrhaging out of the seafloor. Methane and hydrogen sulfide go in, sugar comes out. The clam just sits there and eats the profits. They act like miniature, sealed farming houses, hosting the microbes that do all the actual heavy lifting. Conceptually, it explains how a bivalve feeds without the sun. What it completely fails to explain is how these dense, sprawling carpets of clams can physically exist under 4 tons of pressure per square inch. Visually, their shells look dramatically thinner and more fragile than shallow water clams, almost as if the simple act of calcification becomes mathematically impossible the deeper you go. Building thick armor is biologically expensive, and under this kind of squeeze, the clams seem to have simply abandoned the effort. Instead, they sink their evolutionary budget into chemistry. Scientists have detected specialized molecules that essentially act like biochemical scaffolding, wedging themselves into proteins to physically prop them open so they don't fold up and collapse under the weight of an ocean. They also utilize highly flexible fats in their cell membranes that bend, rather than snap, when compressed. But the larvae remain an absolute mystery. To seed a new colony, microscopic young must survive a horrific, miles-long freefall through freezing thermoclines and crushing pressure gradients, just hoping they land on a toxic chemical seep the size of a dinner plate. Some researchers argue the larvae arrive pre-adapted to the violence of the Hadal zone. Others believe the ancestors slowly adapted in place. We have hints from their genes and their fats. But until we record a full life cycle at depth, these pale ghost farms remain an anomaly. Mecellocephaloides grandifera, a horrific multisyllabic name for a creature that looks like a white, transparent centipede rippling across the mud. Dragging itself between the tube worm towers, it looks incredibly delicate, almost alien. They are long, segmented, and covered in tiny, stiff bristles that anchor them against the deep currents. Subcameras show them swarming anywhere the bacteria carpet the ground. The working theory is that they are the lawnmowers of the deep, mindlessly grazing on microbial mats or scavenging whatever microscopic garbage falls into the trench. That fits neatly into the chemosynthetic model, but it completely ignores the physics problem. These worms possess complex internal organs and muscular systems that, on paper, should fail completely at this depth. Biochemists constantly point to the pressure-friendly fats and the protective piezolites keeping their cells from collapsing. But frankly, evolutionary biologists are baffled by their sheer numbers. Are these ancient indigenous trench monsters or just recent highly adaptable colonizers that tumbled down the slope? We have genetic markers and a handful of lab tests, but zero evidence of how they actually breed and reproduce in a place this hostile. Their ecological role is far bigger than just grazing. Cameras have caught smaller fish aggressively pecking at their backs, and other creatures exclusively nibbling on their waist. They move slowly, but their constant churning of the sediment is what allows the bacteria to bloom in the first place. Lab studies on related species show that they essentially slow every biological process down to a crawl. But if a segmented, highly mobile animal can achieve this level of dominance in the darkest place on Earth, the food web it supports must be staggering. Sea cucumbers. When the rover scans the flat mud, it frequently catches pale, sausage-shaped lumps gliding across the bottom like slow-motion ghosts. They press their fleshy mouths into the silt and mindlessly hoover up the soft sediment, filtering out the microscopic bacteria and whatever organic dust happens to be mixed in. Biologists call them ecosystem engineers. By endlessly stirring and recycling the mud, they act as the biological custodians of the trench. It makes logical sense on paper, but the physical reality of seeing thick, dense herds of them this far down is jarring. Food, by definition, should be mathematically non-existent at this depth. The theories range from an impossibly slow metabolism to the idea that trenches act as geographical funnels, slowly concentrating a global supply of organic garbage into one localized buffet. At a cellular level, the clues are the same: specialized, hyper-flexible fats and stabilizing molecules that prevent their cells from being crushed. But their reproduction remains a total blind spot. We have never watched a deep-sea cucumber larva successfully settle in the Hadal zone. More worryingly, researchers have begun finding traces of long-band industrial chemicals in the tissues of similar trench animals. These toxins attach to sinking particles and inevitably end up concentrated in the very mud these creatures eat. They are the ultimate recyclers, keeping the trench alive by making locked-up nutrients available to everything else. But if remote trenches are already acting as the world's deepest toxic waste dumps, the question isn't just how these cucumbers survive the pressure, the question is how long they can survive us. Snailfish. Floating just above the silt is a soft, gelatinous, pinkish blob. It looks less like an apex survivor and more like a melted tadpole that gave up halfway through evolution. But beneath that pathetic exterior is an absolute masterclass in extreme biochemistry. They are squishy. Their bones are drastically reduced, their skull plates are missing or hyperflexible, and they have completely abandoned the concept of a swim bladder because bringing a balloon filled with gas into a pressure cooker is a terrible idea. Researchers recently isolated a tiny genetic mutation in a gene called RTF1. This single tweak essentially forces the fish's cells to keep reading and executing generic instructions, even when the cell itself is being squeezed to death. They also flood their bodies with trimethylamine N-oxide, or TMAO. Think of TMAO as microscopic rebar. The water pressure desperately wants to fold the fish's proteins into useless shapes, but the TMAO wedges itself into the protein, physically holding it open so the fish can move. Those tricks allow a vertebrate to function where biology usually stops. But the most baffling puzzle remains. How do they find enough calories down here to fuel a predator? Did a shallow water ancestor slowly crawl into the darkness, altering its DNA over millions of years? Or did they arrive already pre-adapted to the cold and just finish the job at the bottom? Scientists can read the genome and measure the chemical rebar, but we still cannot watch their lives unfold. They scavenge crustacean flocks and hunt tiny prey, relying on a strategy of extreme lethargy: move little, grow slow, and live an incredibly long time. If a vertebrate can biologically hack its way into the Hadal zone, it forces us to rewrite the entire textbook on the absolute limits of life on Earth. Giant amphipods. The submersible's light hits something the size of a human fist, its heavily armored legs furiously pumping as it hauls itself across the mud. It looks exactly like a common beach flea, if that beach flea had been exposed to gamma radiation and grown to heroic proportions. These animals are the undisputed cleanup crew of the abyss. They swarm carcasses and pick at the sticky, rotting blankets of bacteria that settle in the trenches. In a shallow tide pool, an amphipod is the size of a grain of rice. Down here, they bulk up into relative monsters. The standard academic explanation points to a combination of freezing water, minimal oxygen, and a complete lack of large predators, which mathematically allows for gigantism. It's a sound theory. What it completely fails to explain is how there are so damn many of them. At a depth where a single meal might drift down once a decade, the math of their abundance does not compute. Researchers have dragged them to the surface inside high-pressure containment spheres and watched them behave completely normally in a lab. The tests confirm they possess the exact same hyperflexible cell membranes and shock-absorbing enzymes as everything else down here. But the supply chain makes no sense. We know trenches act as global sinks, funneling organic detritus into concentrated piles. But does a slow drip of oceanic garbage really explain fields of giant, heavily armored scavengers? Or are there massive, undiscovered chemosynthetic vents quietly fueling an entire hidden economy? When a dead whale manages to sink this far, cameras show thousands of these amphipods swarming the carcass for months, stripping it down to the bone in a frenzy of pure efficiency. Yet, if human deep-sea mining begins to churn the mud and spread the toxic heavy metals currently locked in the sediment, this hyper-efficient recycling system could turn into a poisoned desert overnight. White marine snails. Peppered across the mud between the tube worms are hundreds of tiny, pale shells, looking like scattered buttons. These are not the snails you find in a garden. These mollusks scrape out a living on invisible chemical meals. They use a specialized tongue called a radula to scrape faint, glowing bioluminescent bacterial films off the rocks. Some species even bypass eating entirely, hosting the bacteria directly inside their gills to biologically manufacture their own food. It explains how they survive without a steady rain of surface plankton. But visually, the shells are completely wrong. They are incredibly thin and translucent. Under the immense pressure of the Hadal zone, and with an almost total lack of free calcium carbonate in the water, building a thick shell is nearly impossible. The DNA samples show desperate, highly specialized adaptations in the proteins that form the shell, but it's clearly a biological compromise. They gave up physical armor just to survive the pressure. The biggest missing piece of the puzzle is their children. No one knows how microscopic, soft-bodied snail larvae successfully navigate pitch-black, freezing oceanic currents to magically land on an isolated chemical oasis miles below the surface. Some biologists assume they just ride the slow currents and die by the billions, unless they get lucky. Others think the adults must live for centuries, waiting for the exact perfect moment to release a handful of indestructible young. Because their shells are so chemically compromised, they are incredibly fragile. If the deep ocean mining industry drags a single trawl net through their habitat, reversing the damage wouldn't take years, it would take geologic eras. Dumbo octopus. Out of the pitch-black window, a soft, gelatinous shape flutters past. It looks like a deployed parachute with two large, ear-like fins waving lazily on the sides. It hovers silently above the mud, folds its webbed tentacles around a passing shrimp, and swallows it whole. Unlike shallow-water squid, it doesn't possess ink. Down here, ink is useless. It doesn't possess the muscles for high-speed jet propulsion either. It simply drifts, waits, and pounces. Its body is almost entirely gelatinous, which instantly solves the pressure problem. By matching the density of the surrounding water, there is nothing to crush. The muscles it does have are laced with those familiar bend-don't-break lipids. It survives the extreme lack of food by simply refusing to burn any calories it doesn't have to. The great mystery is why an active, highly advanced cephalopod is so common this far down. Are they migrating massive distances in the dark to find food? Or do they just hover in a state of near-suspended animation, tolerating years of starvation before an accidental meal swims by? If highly intelligent, active hunters can successfully populate the Hadal depths, then the trench food chains are exponentially more complex than our models predict. They hatch from eggs as nearly invisible, buoyant larvae, drifting on the deep currents until they blind luck their way into a food source. They represent a bizarre evolutionary gamble. Invest absolutely zero energy into moving, and just grab whatever happens to float close enough to touch. Lab tests on related species confirm they have some of the slowest metabolic engines ever recorded in a complex animal. But the presence of a predator this advanced begs a deeply unnerving question. What unseen, larger predators are currently hunting them in the dark? Xenophyophores. The submersible spotlight catches what looks like a crumpled, discarded bath sponge sitting quietly on the mud. It is not a sponge. It is a single cell. One solitary cell, grown to the size of a human fist, and sometimes infinitely larger. These are xenophyophores, giant, amoeba-like protists found as deep as 34,800 feet in the Mariana Trench. That specific depth officially makes them the deepest recorded single-celled organisms on the planet. Each one survives by building an outer shell, called a test, by meticulously gluing together bits of sand, mineral grains, and the shattered skeletons of dead animals. The name literally translates to bearer of foreign bodies, and it's flawlessly accurate. They are walking recycling plants built from the garbage of the seafloor. The mechanics of their feeding are simple enough. They extend temporary microscopic arms called pseudopodia and pull in organic dust from the mud. What the scientific community absolutely cannot explain is the biology. How does a single cell grow to the size of a baseball and simply stay intact under pressure that crushes titanium? The liquid inside the cell precisely matches the pressure outside, eliminating the crushing force across the membrane. That physics problem is solved. But physically coordinating a biological body 10 centimeters wide with precisely zero organs and absolutely no nervous system is a biological paradox. More bizarrely, they act as deep-sea chemical sponges. They absorb and concentrate highly toxic heavy metals like lead, uranium, and mercury directly from the water. Instead of immediately dying, they simply warehouse these lethal poisons inside their tissues. No one on Earth understands how a single cell tolerates that specific chemical load. Their ecological role is perhaps their greatest trick. By building these lumpy, garbage-shelled structures, they create physical landscapes on an otherwise completely flat, featureless mud plain. Crustaceans, baby echinoderms, and juvenile mollusks use them as structural shelters. If you remove the xenophyophores, you instantly erase the only nurseries and hiding spots across vast, empty stretches of the trench. Sea anemones. Clinging like bright jewels to the barren rock and the sides of worm tubes, their delicate tentacles pulse gently in the submersible's harsh light. They reach blindly into the freezing water column, waiting to snatch any microscopic crustacean tragic enough to drift past. At Hadal depths, speed is a luxury nothing can afford. They move with agonizing slowness, acting as living tripwires. Many of them also host glowing films of bacteria at their bases, effectively double-dipping into the nearby chemosynthetic energy grids. Because their bodies are entirely soft, flexible, and utterly devoid of gas pockets, the crushing pressure simply passes right through them. The reproductive strategy of a deep-sea anemone is a masterclass in risk. Some species just physically tear themselves in half. Others release a microscopic handful of incredibly large, highly valuable eggs. It's an evolutionary strategy of extreme patience, and it makes them deeply vulnerable. But they aren't just predators, they are architecture. Small, juvenile fish and fragile crustaceans desperately use the spaces between their venomous tentacles as a microscopic safe zone. Erase a localized field of anemones, and you don't just kill a predator, you completely bulldoze a nursery. We have absolutely zero concept of their total lifespan or how far their populations stretch across the trench floors. We just know they exist. If a deep-sea mining operation drags a single dredge across their gardens, destroying the delicate microbial soil they rely on, the damage is essentially permanent. We are currently staring at a profound and ancient ecosystem, and the real mystery is not just how it manages to survive the pressure of the ocean, but whether it will survive us long enough for us to understand it.